So today we are going to discuss chromatin immunoprecipitation. Now this topic is important for GATE, IIT JAM, CSIR NET, DBT-JRF and all the national level competitive exams on biotechnology. So chromatin immunoprecipitation is often acronymed as ChIP assay. So the acronym ChIP is made up of CH, which comes from the first two letters of the word chromatin, and I stands for immuno and pre stands for precipitation. So these four letters, CH from chromatin, I from immuno and P for precipitation, makes the acronym ChIP assay. Okay, now this is widely used in laboratory uses. Okay, now why this is used? This is actually used for figuring out DNA protein interaction. So if we describe it, it is actually an antibody based technique. Okay, why antibody based? Because you will see later on we will use antibody to measure DNA protein interaction. Okay, so it is actually an antibody based technique for figuring out DNA protein interaction. So you can find out where a particular protein of interest binds to which sequence of DNA, okay, by this technique, and then you can quantify it by various sort of DNA quantification techniques. Okay, we will see it later. So it is an antibody based technique for measuring DNA protein interaction. DNA protein interaction means where a particular protein of interest binds to which sequence of protein. Okay, so what do? You first take cells. Okay, you know that nucleus of cells contain DNA. So you first take cells and then you cross-link it. Okay, you cross-link it by the reagent formaldehyde. So by use of formaldehyde, you are cross-linking the protein of interest to the DNA that is present in the cell. So the function of this formaldehyde is it cross-links a particular protein of interest. That means the protein of interest means the protein for which you are required to find out the particular sequence of DNA where the protein binds. Okay, so the function of this formaldehyde is that it cross-links the protein of interest to the sequences of the DNA present in these cells. Okay, any cell you can take formaldehyde. This is the reagent that is used for cross-linking the particular protein of interest to to DNA. Suppose this is the sequence of DNA that you have. Okay, and this is the protein of interest. Okay, and you are required to bind this protein of interest to this sequence of DNA with the use of formaldehyde. So you cross-link the protein of interest to the sequences of the DNA with the help of this reagent formaldehyde, and then you perform cell lysis. Okay, then you lyse the cells. You can use various sort of lysogenic agents. Okay, if it is bacterial cells, you can use lysozyme. It is fungal cells, you can use cellulase, like that. Cellulase chitinase. Okay, so you can use various sort of lytic agents here for lysis of the cell walls and the cell membrane. So you can use various sort of lytic agents, lytic reagents, which perform cell lysis. Okay, to lyse the cells and isolate the DNA, or here it is called chromatin. Chromatin is actually a condensed form of DNA. So here you isolate the chromatin by cell lysis. Okay, and then here you can see these these green ones are actually the protein of interest which you have cross-linked with the use of formaldehyde. So these green ones are actually proteins. These are actually what? Protein of interest which you have cross-linked with the use of the reagent formaldehyde. Okay, so you can see these proteins have bind to the chromatin, to places in the chromatin, and these pinkish ones are the chromatin. Okay, these pinkish ones are the chromatin, and here you can see the proteins have bind to the chromatin. Now what do? You perform sonication to break this whole chromatin into small sets of chromatin. Like here it will cut, like then here it will again cut, like here it will again cut. So you break it into small pieces, each of 200 to 400 base pairs. Okay, so the sizes of this small chromatin pieces after sonication must be between 200 to 500 400 to 400 base pairs. Okay, so you sonicate and you break down the whole chromatin with the bound protein into small pieces of chromatin bound to protein. Some are bound to protein, some are not, and the sizes of those small chromatin pieces are between 200 to 400 base pair, and then you immunoprecipitate it with antibody attached to a bead. Okay, so this is the bead. This is the bead. It is here you can use two types of beads. Either you can use agarose beads. Okay, mostly agarose beads are used for immunoprecipitation, but nowadays some researchers have begun to use magnetic beads. Okay, so you can use either of the two types of beads, either agarose beads or magnetic beads. And first you fix the antibodies. So these are actually the antibody, fixed antibodies. Okay, these are actually the antibodies. So you fix the antibodies to the agarose beads, and then you allow to make these antibodies react with this small fragments of protein bound chromatin. So what you will observe, this antibody is actually binding to the protein of interest. Why? Because these antibodies have been made in such a way that they are sort of complementary to the protein of interest, which are already bound to the chromatin. So only those pieces of chromatin which have bound proteins with them will bind to those antibodies. Otherwise, these pieces of chromatin, here you will see, here this piece of chromatin has no protein with it, associated with it. So this piece of chromatin has no protein associated with it, so it will not be able to bind to the antibody, which is specifically designed such that it is complementary to the protein of interest. So these two fragments will not be able to bind, and it will go out earlier. Only those pieces of chromatin which have proteins associated with them will be able to bind to this antibody. They will stick to this antibody. Okay, this is called as bead antibody protein target DNA complex. So once this complex is formed, what do? You perform protein digestion. That means you perform reverse cross linking. Okay, you perform reverse cross linking, or you perform protein digestion to remove the bound proteins that you have. Okay, to remove the bound proteins that you have. Okay, and now you take out the DNA, and now you isolate DNA, you purify DNA, and then you can perform DNA analysis by either PCR, okay, either PCR or by qPCR. So this is the first method, this is the second method, or by microarray, this is the third method, or by automated DNA sequencing. This is the fourth method. So you can perform chromatin immunoprecipitation. In chromatin immunoprecipitation, you can perform DNA analysis by either of the following methods, either by PCR, either by qPCR, either by microarray, or either by automated DNA sequencing. Right? So this is basically, now the question arises, why will we use chromatin immunoprecipitation? Okay, so we use chromatin immunoprecipitation mainly to find out the target sequence where a particular protein of interest is binding. Okay, so by PCR, okay, you can see by PCR what happens. Only those portions of the DNA which will bind to the protein of interest, only those portions will be amplified. See here, if I choose another color, see here, when you are performing sonication, okay, here the protein is binding to the this piece of chromatin, so here you will not have any fragmentation. Here you will have a fragmentation because there is no protein binding here. Again here the protein is binding to the chromatin, so here no fragmentation. Okay, so here no fragmentation. Again you will have fragmentation. Again you will have fragmentation here. Again here you will have no fragmentation because a particular protein is binding here. So only those portions of chromatin where you have a protein binding, you will not have any fragmentation. Other portions where you don't have any protein binding to them, you will have fragmentation. And also you can easily see here, only those portions of the chromatin which are binding or which have bound proteins associated with them will be able to bind to the antibodies, right? Here you can again see that only those portions of the chromatin which are able to bind the proteins will be able to bind to the antibodies. So other portions where you don't have a protein binding to the chromatin will not be able to bind to the antibody, and they hence will be removed. So here, because of these two reasons, first of all, the portions of the protein where the protein is binding to the target DNA, those portions you won't be able to perform fragmentation. And second of all, because those portions of protein where the protein is binding to a particular sequence of DNA, only those portions will be able to bind to the antibody, other portions will not be able to bind. So once you have figured out which chromatins are binding to the protein, you perform reverse cross-linking, or you perform protein digestion to remove this protein, and then you take this chromatin, you purify them, and then you perform PCR analysis. Okay, you perform PCR analysis and you amplify those DNA segments by means of a forward primer, by another you have a reverse primer to amplify the sequence of the DNA, and then you can see which sort of DNA sequences are binding to that particular protein of interest. So this ChIP assay has more or less 5 basic steps associated with it. So first step is performing cross-linking. So this is the first step where you perform cross-linking. Second step is performing chromatin fragmentation by means of sonication. So you perform cross-linking by means of formaldehyde, and second step is performing chromatin fragmentation by means of sonication. Okay, so this is the second step that you have. Chromatin fragmentation. The third step that you have is immunoprecipitation with antibody complementary to the protein of interest and using agarose or magnetic beads. So third step is immunoprecipitation. Okay, so this is the third step where you immunoprecipitate it with antibodies and you use beads. Okay, so this is the third step. Fourth step you have DNA purification. That means you digest the protein, you reverse cross-link the protein, and you remove the protein because the function of protein is now over. You have found out which particular DNAs are binding to that protein. Your next objective will be to find the sequence of the DNA where the protein is binding. So this is the fourth step. And how do you perform or how do you find which particular DNA sequence is binding to which protein? You perform PCR, right? So this is the fourth step where you perform DNA purification. Okay, you find out which particular DNA which particular DNAs are binding to that protein of interest, which fragments of DNAs rather, and then once you have found out which fragments of DNA are binding to that protein of interest, the function of protein is now over, the role of protein is now over, and then you purify the DNA, you digest the protein and purify the DNA. Your next step is to observe what is the sequence of the DNA fragments which are binding to that particular protein of interest. So the fifth step is DNA analysis. Okay, so DNA analysis, you can perform this by either PCR, okay, by either qPCR, you can observe it in real time, or you can perform microarray, okay, or you can perform automated DNA sequencing. So these are the 5 steps by which chromatin precipitation can be performed. The main purpose of performing this ChIP assay is to find out which particular protein of interest is binding to a particular sequence of DNA, and to find out where that protein is also binding. So here we are answering to 2 questions: whether a particular protein of interest is binding to a sequence of DNA or not, and if they are binding, where they are binding. We have to find out the sequence also, so that it can be standardized and we can say in future, so this is the DNA sequence which always binds to this particular protein of interest. That is the ultimate goal of performing ChIP assay.